these are the steps that are all accomplished through the action of different enzymes, just like with DNA replication. So we have to first bind to the DNA and unwind it or split apart those strands. Then we have to begin synthesis. So usually in chemical or metabolic processes, there's some special step for the very beginning, just like with DNA replication. Yeah, and we'll go through all these steps. So binding and unwinding, initiation, elongation, which is just adding those nucleotides, or adding those monomers, and then termination or stopping. So I have a little video that I'm going to show. And this is linked in the slideshow. There are a couple. I linked this one as well as another one. They're both pretty good. They're a little bit dry, but you know what? A lot of the other videos I was finding online for Khan Academy and stuff just went off, and they didn't just talk about transcription, they talked about a lot of other things. So I wanted to just get the basic information for transcription in there for you.
Transcription of a gene or group of adjacent genes called an operon is controlled by a set of proteins interacting with specific sequences in the DNA molecule.
Okay, so just to start off, they're referring to just a region of DNA, and we'll talk about what an operon is in a couple of minutes, but this is talking about bacterial transcription, which is a lot more simplified than eukaryotic, so we'll talk about that first, and then we'll talk about eukaryotic. So I'm just focusing on the major steps here.
Leading to the initiation of transcription and the termination of a transcript. Transcription is initiated just beyond the yellow promoter region and is terminated at the green terminator site. The sequence of the promoter at positions 35 and 10 nucleotides upstream from the transcription start site are critical to initiating transcription. In order to initiate transcription in bacteria, core RNA polymerase and sigma factor form a holoenzyme. Sigma factor recognizes the sequence of DNA at the promoter region.
Okay. Okay, well, process for me, because this is one of the most important parts that I wanted to cover. So, as we said, one of the major steps in transcription is initiation. And initiation is when the RNA polymerase binds to the protein base on the DNA, and it needs to be positioned correctly to say, go this way or go that way. That job is done by this other protein. In bacteria, it's called sigma factor. So this is the sigma factor. The sigma factor actually is what tells the RNA polymerase what direction to go in. And it's a little bit different in eukaryotes, but in prokaryotes, it's just up to this one protein. So let's go back to the slideshow, actually. That's more or less. That was the most important thing that I wanted to show you. And then after that occurs, then we have sigma factor leaving and elongation and termination, which we'll talk about in just a moment. So down goes the links. So the enzymes that we need to know for these processes are RNA polymerase and sigma factor, which we just saw. And then the parts of the DNA are the promoter and the gene sequence. And we saw pretty much all of that in that, 20 seconds in that video. So again, we're starting out talking about prokaryotic transcription. It's a lot simpler. What is a prokaryote as opposed to a eukaryote? Yes, so prokaryotic cells or organisms don't have membrane-enclosed nucleus, they don't have a membrane-enclosed endoplasmic reticulum, anything like that. They just have their outer membrane. DNA is inside of the cell, not enclosed in any particular organelle. And these are bacteria and archaea. And then things that have a nucleus are eukaryotes. So the first step that we saw in the video was that RNA polymerase, again, our polymerase enzyme, which is going to be responsible for catalyzing the formation of phosphodiester bonds, or in other words, RNA polymerase is going to be responsible for adding nucleotides into this string of nucleotides. It first needs to bind to this other protein called a sigma factor. And in this image, it's shown a little bit differently than in the video, but we still see the two pieces. So we see in blue here, the large part is RNA polymerase, and then the small circular part is the sigma factor. Again, the sigma factor is not going to be in charge of catalyzing any reactions. Its purpose is to find the beginning of a gene and point the RNA polymerase in the right direction. So imagine this. Think of yourself being an RNA polymerase trying to find a specific gene to transcribe. You guys know I like to anthropomorphize a lot. So the genome is really big. Where do you start copying DNA into RNA? And what direction do you go? Even if you know you're at the beginning of a gene, or is the gene in front of you, or is the gene behind you? Because if you go in either direction, you have a double helix, right? So the RNA polymerase isn't in charge of that. That's what sigma factor is for. Sigma factor finds the start of a gene, and it says, okay, go straight forward in this direction. That's where you want to start copying and making the RNA. So if the RNA polymerase is not bound to sigma factor, it's not going to find the beginning of a gene, and it's just going to kind of wander around. It's never going to actually start transcription. So as I said a minute ago, we have two strands of DNA, and they are anti-parallel. So this image shows how you have genes going in opposite directions in the same gene. So on the left here we have gene A, on the right we have gene B. So gene A, if you're going to read it, which way do we read? Read it right? So we're going to read gene A. Go from the 3' end towards the 5' end. So that means we need to use this bottom strand. If we're looking at gene B, so that this is gene B, start is here, we need to read 3 to 5. We go from 3 to 5 from the top strand that we need to read. We'll get some practice with this in a little bit. So the important things to know for which strand you need to use to make a gene or to transcribe a gene are where the promoter is, so where the start is, and where the gene is. And also, of course, 5' and then you can figure out which direction you need to go. So again, if you know where the promoter is, the starting part, then you know that you can always go from the promoter towards the 5' end, and that'll tell you which strand you need to use. And this is just a real example that I pulled out of an example of the human gene sequence. And we have many genes close to one another in the genome. And it might be kind of hard for you guys to see in the back, but there's one gene here in purple, and then there's another gene here in purple, and the gene on the right has arrows going this way. And the gene on the left has arrows going that way. So these are two genes really close to each other in the human genome that are read in opposite directions. And this is found in all organisms, so far as I know. So the sigma factor is what tells the RNA polymerase, hey, there's a promoter here, and you need to go in this direction in order to transcribe the gene. And it does that by looking at the promoter. So the promoter is a region that is at the beginning of a gene. It's before any of the nucleotides actually code for any amino acids for the protein. First part, and nucleotides only start coding for protein sequence after the promoter. First the 0 position, and then we'll have 1 remaining nucleotides we have. And we'll do something like that. Polymorph. And within this promoter sequence for bacteria, it's called -10 and -35 sequence. So these are just numbers with positions of nucleotides. And they have particular sequences which the sigma factor protein can recognize. So again, this all comes down to chemistry. The sigma factor recognizes these sequences. What we really mean is that it chemically interacts with these sequences so that it arranges itself in a certain way. So that's why I really liked this video that I attempted to show you, because it's showing here, it's not yet aligned in this image, but you can see that the sigma factor is kind of oblong shaped, and it has two different ends. And so this end would align with the -35 part, and then this end would align with the -10 sequence, so that we've created a direction, basically a molecule that's an arrow, more or less. It's saying, okay, go down this way. So once the sigma factor is aligned correctly with the -35 and the -10 sequence, then the RNA polymerase recognizes that it's at the start site. And we'll see here that the start site, this is where we want to begin transcription, because this is where the nucleotides start coding for amino acids. So once the whole complex, the sigma factor and RNA polymerase are at that start site, then RNA polymerase binds tightly. Before that, RNA polymerase was not bound tightly. And I think that's why they shows this kind of diagram. They're showing that sigma factor is kind of in the way. So while sigma factor is there, the RNA polymerase is not going to bind tightly to the DNA. And the other textbook that I used, it refers to RNA polymerase just sliding down the DNA strand. So I use this. Sliding down the DNA strand, it's not binding tightly. When it gets to that point, where it is here, the sigma factor finds the promoter and it aligns itself with the -35 and the -10. Then sigma factor leaves. And that's when RNA polymerase kind of clamps down or it binds more tightly to the DNA and it starts transcription. So again, in this image that we're seeing, in this version, when sigma factor's there, it's kind of in the way, and RNA polymerase can't bind as tightly to the DNA, but once we get rid of sigma factor, then RNA polymerase binds tightly and starts actually catalyzing reactions. Yes, this is very fast. And so that's a good question. That's part of why I mentioned this step. Because we're thinking of it with intentionality, but there are RNA polymerase molecules all the time that are attached to the DNA. And you might have a sigma factor, and they're just moving along the DNA. So they're not actively transcribing, but then when the conditions become right, and they come across a particular start site, then the sigma factor will leave, and then we start transcription. So everything's kind of always in place for transcription to happen in terms of the RNA polymerases. And we'll talk more about things that might stop that in tomorrow's class. Yes, okay. Which one was it? Can you tell me what year?
This one's about to do is this one and the spreadsheet that we have on the slides is already good. It's slides not really showing.
Oh, yeah, so then I apologize, I'm remembering this different. So the order is that we have RNA polymerase, and I'll upload these output files. RNA polymerase plus sigma factor, and this is when it's sliding or searching for that start. And then together, once they hit the start site, then signal leaves. And then, because again, we're getting that sigma factor out of the way, so to speak, RNA polymerase binds tightly and transcription begins. And this is at the start site. And this is does that clarify? No. Good. All right. So then we just have RNA polymerase making RNA. So it's going through this process of reading the DNA template and then creating this strand of RNA. The process is very similar to DNA replication. We're going to read a template, form phosphodiester bonds, and the synthesis direction is 5' to 3'. And let me go back a slide or two just to give us. So the naming of this can be a little bit tricky. So there are 2 strands of DNA. As we've said, you use only one of them for any given gene. You use only one of them for transcription. So that would be the template strand. The other strand of DNA that RNA polymerase is not reading is called the coding strand. So you see up here, the top is the coding strand in this example. And the coding strand is, again, the one that RNA polymerase does not use. And because of the complementarity of DNA and RNA, the coding strand is actually exactly the same as the RNA that's going to be made, except for its DNA instead of RNA. And we see that here. So if we look at the 5' end of the coding strand of DNA, we see APG. We look at the 5' end of the RNA that's being made, AUG, so they're the same. So both the coding strand and the RNA are complementary to the template strand. And again, remember that for this gene, the top strand is the coding strand and it's not really useful. For the next gene in the genome, the coding strand might be the bottom strand. It depends on where that promoter is. So how do we actually make the RNA? Again, really similar to DNA replication, where we have this polymerase that is taking nucleotides, matching them up to the template, hydrogen bonding, and then forming phosphodiester bonds to add them to this growing chain. And really similar to DNA replication, we have the same process. The nucleotides that are incoming are triphosphates. Why do we need this triphosphates? So we remove 2 phosphate groups. Energy? Yeah. They generate energy that fuels the reaction, and then the 3rd phosphate stays on to become part of the sugar phosphate backbone. So now we make RNA, we transcribe the gene, and at some point we have to stop transcription, right? So that occurs when the RNA polymerase hits a certain pattern or a certain DNA code, which is called a termination sequence. And then it just falls off. And then what is this image showing? After, as soon as the RNA polymerase stops transcription, again, it can't take a break. It binds to a new sigma factor, and then it rebinds the DNA and starts transcribing another gene. But at this point, we have the mRNA gene that we just. So similar to other things that we've talked about, the terminator sequence has a particular composition of DNA. Often it's high in A's and T's, and it causes certain conditions so that the RNA polymerase is going to fall off. Other thing that I wanted to talk about, which is related, is what an operon is. Does anyone know what an operon is in genetics? So operons are only found in prokaryotes. We don't have operons in eukaryotes. But operons are areas on the chromosome or in the genome that have sets of genes that are all used for the same process. So a really good example of an operon is the lac operon. It's for lactose. It has genes for all kinds of things related to lactose, and you'll see that later. But basically, it's keeping genes that are related together. So we have gene 1 here, and we have gene 2, and then we have gene 3, and that's in the prokaryote. And these guys, they're always expressed together because they have a common overall function. So they have a single promoter, and they have a single terminator. And this results in an mRNA that contains 3 different genes. And again, we'll come back to this later. The important thing here again is you're getting 3 different mRNA sequences all attached to one another. Single chain, continuous phosphodiester backbone. And Again, this is because these 3 proteins are going to be used in the same process. So it's useful to have them transcribed at the same time.
Are there any questions
About transcription. So which one is that? This one? This is just a continuation of our performance. Adding up. This one? This one? So we're gonna take a break now, and I will upload this copy of this PowerPoint. And I apologize that that was not. Forgot that. Unless anybody has any other specific questions about transcription. Exactly. Yes. And so we'll go back to this, and we're gonna work through it. Okay, so let's take a 10-minute break, actually. This is a lot, and we have a lot to talk about for eukaryotic transcription. Just lecture. Yeah. Yes. And no, just read over the protein this on 4. Now that the internet is actually working for me, let's finish watching this video since it's pretty quick. And this is the video that's linked in the
Core RNA polymerase and sigma factor form a holoenzyme. Sigma factor
I just wanted to note also, so holoenzyme is a term that you might see. It just means that it's an enzyme that has more than one part.
recognizes the sequence of DNA at the promoter region. This holoenzyme then binds to the promoter region of the DNA molecule, forming the closed complex between RNA polymerase and the DNA molecule. By breaking hydrogen bonds between bases in the double helix, the closed complex is converted into the open complex. The formation of the RNA transcript from nucleotide triphosphates then takes place as RNA polymerase is forming the new transcript, starting at the 5' end of the transcript. New nucleotides are added onto the free 3' end. The initiation of transcription is completed when sigma factor is released. As elongation of the transcript then continues, the RNA polymerase moves down the DNA molecule and the next sequence in the chain is opened up. As RNA polymerase moves down the DNA strand, the DNA molecule entering RNA polymerase unwinds, forming the open complex, while the DNA exiting RNA polymerase rewinds to form double helix. Ribonucleotide triphosphates are added to the chain, and the growing RNA transcript continues to elongate as new DNA sequence enters the open complex region. The new RNA transcript initially forms an RNA-DNA hybrid, complementary to the template strand, corresponding to the sequence of the coding DNA strand. In this way, the DNA double helix is opened, transcribed, and reclosed with minimal stress on the DNA molecule. One mechanism of bacterial transcriptional termination involves the binding.
So we'll stop there. First, if you're watching this on your own, I would stop there as well. It starts talking about other proteins that are not universal and that we don't need to know. But I do want to point out that this video really emphasizes that only a small portion of the DNA helix is open during transcription. And that's different than DNA replication, right? Where we have to open a large replication bubble, and then we use things like topoisomerase to relieve the stress in other areas of the DNA molecule. Here we don't come across that problem. We're just pulling apart a few base pairs at a time, and then we're allowing them to come back together. I also want to point out that notice that there's a brief DNA RNA hybrid that's formed as we're reading the template DNA and matching up RNA to that. We see that here, and there are a couple other places where we're going to see that. I just point this out because if you go into medical fields, you might come across back to that DNA, RNA hybrid molecules can sometimes cause what I would call a cellular immune response. So sometimes there are certain molecules that we recognize them and destroy them because that can also be part of a viral life cycle, having the DNA and RNA together. So we do have that here, but it's very short-lived.
That's good.
No. And I did post now an updated slideshow. Again, I'm really sorry that I did not post that. So now we're going to talk about eukaryotes. And you're going to see why we talk about eukaryotes first, because they're much more straightforward. And they're more consistent within the group of prokaryotes. Eukaryotes are all over the place. I would say that eukaryotes are much higher maintenance than prokaryotes. I would be a bacteria. In eukaryotes, we have a bunch of different proteins that are in charge of initiation. We're not going to learn every single one of them, but you should know that there are a number of them. The structure of a gene in eukaryotes can also be different than prokaryotes. And we'll talk about this in detail later on. The overall theme though is the same. We're going to see that there's an initiation process, and then there is a start of transcription. We're adding on nucleotides to this RNA transcript, and then there's some kind of termination. But then, after all that, which we have in prokaryotes, there's going to be something called mRNA processing, which is crucial for eukaryotes because of the fact that they have a nucleus. So, one of the first differences between eukaryotes and prokaryotes is the promoter itself. So in prokaryotes, we said that the promoter was at a specific place upstream of the gene. We know it's a specific place because we call it a -10 and then -35. So it's always 10 base pairs ahead of the start site and then 35 base pairs ahead of the start site. Those two pieces always in the same place. In eukaryotes, the promoter is not always in those specific positions and is not always a particular sequence. But we refer to it as the TATA box because it often has TA, TA, and sometimes repeats of that. So we refer to eukaryotic promoters as TATA boxes. And again, that would be analogous to the -10, -35 sequence in the prokaryote. Another difference is, of course, the actual enzyme itself. So RNA polymerase in eukaryotes is different than in prokaryotes. And actually, in eukaryotes, we have 3 different RNA polymerase enzymes. They're all similar, they have slightly different jobs. Just the way we saw that in eukaryotes, we have different DNA polymerase enzymes. The main difference here is which genes these RNA polymerases are most commonly transcribing. And RNA polymerase 2 is our main RNA polymerase. It's going to be transcribing most genes that are protein coded. So basically, whenever you have a gene that's encoded for a protein, you're going to have RNA polymerase II making that mRNA. Then we have these other RNA polymerases that we're not going to talk very much about, but they actually make RNA that stays as RNA. And we'll talk more about that tomorrow. So as we said, eukaryotes have a top-down boxes that say 10 and 5 sequences. These are both referred to as promoters. And I want to point out this link.
It's 11 o'clock.
Are promoter sequences upstream or downstream of a gene? So you know, someone guess. Are promoter sequences upstream or downstream of the gene? Promoter sequences, whether you're talking about eukaryotes or prokaryotes, are always what we refer to as upstream of the gene. So what we mean is if we have a gene here, and we know that this gene is read in this direction, for example, we have to go upstream against the flow here, and the promoter is going to be here. So we can also have a gene going in the opposite direction. In this case, the promoter is going to be here. So we have to go against the opposite direction of how you would read the gene. The promoter is where the RNA polymerase starts. So that's always upstream. Yes. Yeah. And then downstream of the gene refers to things that are after the termination cell. Downstream is after the protein. So downstream is going to be, so if you're reading a gene, opposite synthesis, downstream would be 2 of the 5 of the 10. Right, so the promoter would be at the 3' end of the template strand. Because, again, we always synthesize 5' to 3'. So we know that we're going to start synthesizing, starting off with the 5' end from the transcript. So in order for that to match up with the template, this is the RNA. And we run 3' to 5'. The promoter is at the 3' end. The terminator is at 5' end. Upstream is 3'. downstream is 5 prime. But if we look at the complementary strands, so this is the template strand, what's the other one? The coding strand. The coding strand is going to be opposite. So you really have to pay attention to which strand you're talking about. And this is, and so the direct now is reversed. So in this case, we're talking about the coding strand that the downstream end of the piece. And we wouldn't normally refer to it that way. Just keep in mind that just because you see a 5 prime end, it doesn't necessarily mean it's downstream. You have to figure out which direction the gene is going. And that'll become clear when you actually work on problems with this. Right now, this just sounds like a lot of details. We'll come back to this when I have a worksheet. I challenge you to come back to this. Okay. So let's talk about a bunch of different proteins first, and then we'll talk more about directionality. So as we were saying, the promoter for eukaryotic genes is a Tata box. Instead of a sigma factor, we have a different protein. This is called transcription factor TF to H, and transcription factor 2D, which has a special component, which is called TEP, for Tata binding protein. So this protein that binds the Tata box is called the Tata binding protein. Just to be extra, the Tata binding protein. So that's the protein that actually binds, start the motor, and begins this whole process. And as we saw in prokaryotes, or what we saw in prokaryotes, was that there was just the sigma factor that localized there, and then brought in the RNA polymerase, where they went there together, and then the sigma factor recognized. In this case, there are a whole bunch of other proteins that come in before RNA polymerase comes. So these are called general transcription factors. General transcription factors are just a bunch of proteins that assemble around the Tata box during transcription initiation. So again, a lot of different proteins that assemble around the Tata box during transcription initiation in eukaryotes. So these are called general transcription factors because regardless of what gene we're talking about in a eukaryote, they all need this set of like 12 different transcription factors or 12 different proteins. And again, this is why I said that eukaryotes are high maintenance. Their gene transcription is just like, just like divotastic or something. We have all these different proteins. Bacteria, we just had sigma factors, so simple. Transcription factors. Yes, and so that is something we'll get to later on. Right now, we're just talking about general transcription factors that associate with the Tata box. And so I know this is not an image from your textbook, this is another textbook, but I use it just to illustrate how there are all these different proteins The first one, importantly, is the one that carries the TATA binding protein. So that actually starts the kind of process of gathering all of these proteins. But once that is there, this protein that has the TATA binding protein, then all these other proteins start coming to this area. The RNA polymerase also joins to this area in this whole gathering process, this initiation process. And I think that this is a good image right here in the middle. So we have a bunch of proteins. They're all associated or gathered around this green area here, which is the TATA box. We also have the RNA polymerase, which all these proteins, again, just like the sigma factor, they're kind of stabilizing it, they're putting it in the right place, and they're going to send it off to start transcription. So that's the point of all these general transcription factors, is to bring the RNA polymerase to the TATA box, so that's going to start transcription. The signal for RNA polymerase to actually start transcription is actually being phosphorylated. So there are certain general transcription factors that will add phosphate groups to the RNA polymerase. And again, when we add phosphate groups, what does that do? It can activate or deactivate, and in general, it changes the structure. And so this changes the structure, changes the shape of RNA polymerase a little bit so that it binds tightly and it's ready to start transcribing. So at that point, most of these general transcription factors then leave. And so students always say that what was the point of all those transcription factors? And it's a little bit hard to really understand and try to justify to yourself why do they all assemble. So you have to imagine, again, there's all this chemistry going on, these 3D shapes fitting into each other so that this assembly is a 3D assembly, it's fitting together and it's stabilizing the RNA polymerase to its correct position and getting it ready to start transcription. So that's the importance of that. And the final step of preparing the RNA polymerase is to phosphorylate it and then it can start transcription. Yeah. General transcription factors and GTF, everything in position, and then finally, one of them, or a few of them, add a phosphate group to the RNA polymerase, to specific places on the RNA polymerase, changes its shape, and then starts transcription. Any questions about this? I also forgot now that I did put on slides for this slideshow, and I'll continue to do this for other parts of the semester. Eukaryotic exclusive seal, and that means that this only happens in eukaryotes, so that if you're confused about whether this is eukaryotes or prokaryotes, you would always look to that. So speaking of the difference between eukaryotes and prokaryotes, now that we've started transcription and we know that the process of actually making RNA is essentially the same for RNA polymerase in prokaryotes and eukaryotes, and termination is also very similar, then what else do we have to talk about? Well, after the mRNA is made, it's very different for prokaryotes versus eukaryotes. So the mRNA, actually, it's not strictly after the mRNA is made, but even as it's being processed, other enzymes start to modify this chain of ribonucleotides. And there are a couple of different purposes for this. One of them is that we need to have modifications in order for this mRNA to leave the nucleus. Another reason for this is something called splicing. Does this ring a bell to anyone? Yeah. So, as I said, eukaryotes tend to carry around more DNA than prokaryotes. And in part, this leads to genes that are not as kind of efficient in some way or compact as prokaryotic genes. So in eukaryotic genes, we might have different regions interspersed into the gene that are not actually going to be used in making a protein. The final messenger RNA transcript is shown here, but this is the DNA or initial RNA transcript here. And so these red pieces need to be taken out. And these guys are called introns. And these are pieces of, or these are sequences that are not going to be part of the final mRNA. And I'll talk a little bit more about them in a moment. But I want to first give us just an overview of what we're talking about. So, mRNA processing has three different components: adding a 5' cap, and also adding 3' tail. So adding things on each end of the mRNA, and then in the middle, we have splicing. Again, all of these things contribute to the correct mRNA being made that can be used by the ribosome to make the protein, and allowing the mRNA to actually leave the nucleus. It's pretty important because are there ribosomes inside of the nucleus? No, and no ribosomes in the nucleus. So that's a really important detail that sometimes gets lost. But we cannot make proteins unless we could get the mRNA out of the nucleus into the cytoplasm. So a moment ago, we were talking about certain general transcription factors, adding phosphate groups onto the RNA polymerase. So we said that that changes the shape a little bit and allows RNA polymerase to start transcribing. The phosphate groups also act as signposts, kind of, for some other proteins that are going to localize to that area, and they're kind of waiting for the transcript to be made so that they can start processing the mRNA. And so these are the enzymes that are responsible for adding the 5' cap, the 3' tail, and for doing the splicing. So this is what mature mRNA looks like. You see that it has a 5' cap. And so the 5' cap is actually just a modified nucleotide, a modified G, and then the 3' tail, the poly A tail, is also nucleotide, just a bunch of A's. So that's what's on either end of the mRNA. And then in the middle, as we said, we only have the protein sequence, protein coding sequence of the gene. There might be some non-coding sequence, but this is not considered an enzyme. So we only have exons in the mature or processed mRNA. That's where the lead nucleus. So here we're just talking a little bit about why these things are needed. But as I already said, the black prime and the blue prime are really important for getting this mRNA transcript out of the nucleus. And also actually important for beginning translation, which is the next step. And then splicing. Splicing is important because, as we said, eukaryotic genes tend to have a lot of regions that don't contribute to a protein. Or eukaryotic genes have kind of a mix and match situation where you don't need to use all of that sequence to make a particular form of a protein. So we've talked about different structures, different segments. The helices, the sheets, you have multiple of these in a protein. So imagine a protein that's very large and has 10 beta sheets that are stacked up. And for this gene, we'll say that when it's transcribed, initially, the unprocessed mRNA has these portions that code for all 10 beta sheets. But perhaps all 10 of them aren't needed in all situations. So this will be processed, and some of these will be removed. So we can mix and match. You don't need to keep all 10 of them in. We can have 5. And this is not random, obviously. There are different enzymes that are regulating and dictating how this is processed. But this would be a good example of mix and match. Usually, this is explained as there are exons which stay in and are important and code for protein, and then there are introns which are not important and they don't code for protein. But there's also kind of a gray area where there are certain exons that will only use something specific. And we'll see this in one of the problem sets when we do this. And so what happens is that we're actually going to break the phosphodiester backbone of the mRNA to piece together all the exons which are going to stay in the mature mRNA. I think because it was made in French. Yes, yes. So it's, you can remember it as the opposite. You can remember it as the exons are allowed to exit the nucleus. I have student came with another one I can't remember. If you guys come up with anything, have a good way to remember that. Please share. So this process occurs by what they call a spliceosome. And the spliceosome is what does the splicing. And it actually, again, cuts that phosphodiester backbone, and it takes those introns right out. And then it ligates the phosphodiester backbone. Splice, yeah. Yeah, it has an element. Let me fix that. spliceosome is again what recognizes the introns, removes them, and then ligates the exons together. And importantly, something that we didn't really know about until a couple of years ago, maybe a little bit more, but RNA is a big area of research right now, in part because of this. So the spliceosome recognizes the introns that it needs to remove because there are actually complementary sequences of RNA in the spliceosome. So you don't need to know all the details of this, but I have seen these words in the news sometimes. So just to connect with that, if you've heard of small nuclear RNAs or things called SNRPs, you've heard of these on the Science Channel and stuff. So this is what they're talking about. It's part of the spliceosome. And what you should know is the introns do have short sequences that code for their own removal. It is based on sequence. So if you change those sequences in the intron, then you could mess up the protein, even though the intron shouldn't be coding for amino acids, right? Because if you change the sequence of the intron, then it might not be removed. And then it would be read during translation, and that's bad. You don't want that. So if you had a mutation in your DNA, or even if we had incorrect transcription in the portion of the intron, well, intron, like normally you say, oh, it doesn't matter because it's not coding for amino acids. But if you change that sequence so that the intron is not removed, then the final mRNA is incorrect. Yes, so that would cause a mutation that would commit exactly the protein. Now, the other thing here I want to point out is that introns are removed before the mRNA leaves the nucleus, and they're going to be degraded. Those ribonucleotides are going to be recycled, and they're just used for another transcription initiative. So again, the conservation of materials is important in this. Okay, so once this mRNA is all processed, we've added the 5' cap, we spliced, and we added the 3' poly A tail. Then it goes through these nucleophores, which we've talked about before. Again, it needs to go through these areas of these unstructured regions that only certain proteins can get through. And they're able to do this because there are certain proteins that recognize 5' cap. There are certain proteins that recognize the poly A tail. And after splicing, there are certain proteins that stay with the mRNA. So these three types of proteins help this mRNA get through the nucleus. That's another reason why mRNA processing is really important. And once that happens, then it's going to be in the nucleus and just about ready for translation. So are there any questions? What can we go back to? Sorry, I'm flying. Let's talk about, let's circle back to this. Referring to this being the upstream and downstream.
So when we started at the kindergarten, when they told us there was a specific concept after that, like where it started with slopes and
Oh, good question. So no, the process would be completely different because only RNA compares to makes messenger RNA and one messenger RNA is plasmids.
so that's when we were at MXCC.
So the tRNAs can also be the nucleus, but we have a different target process. Any other questions about this? Okay, so talking about upstream versus downstream. So if we have a DNA sequence here, and we have our two strands, they're anti-parallel. The coding sequence is on where the template strand is the top strand, and then the promoter here. This region is upstream of the promoter, if the gene is here. This region is downstream of the gene. Upstream of the promoter, again, is usually, or upstream of the start site is usually indicated by negative numbers, because it's before 0 for the start site. So again, the promoter is close to the 3' end, and the end of the gene is close to the 5' end. It's opposite for the complementary strand. So if you're looking at this region here, this is upstream of the promoter, but if you're looking at the opposite strand, you'll see that it's closer to the 5' end, so that could become confusing. So you really need, when you're referring to upstream or downstream sequences, you need to know which strand you're talking about, and it's with respect to a particular gene. Because again, as we said, there could be another gene right here. Doesn't overlap. And then this would be the upstream region for this gene. And this would be the downstream region. We see that it stays consistent in that for this gene, the upstream region is at the 3' end, and the downstream region is at the 5' end, if you're looking at that template string. Did that clarify a little bit?
Is that negative 100? What is that?
So this, the number 1 is the first nucleotide that codes for, is part of a codon. So the first nucleotide that is part of a codon that will code for amino acids. Yes, so number one would be A in the start site. So start. Is AG, and so the one, or the A would be one. And then you just count backwards from that.
So this should be this would give us the points correctly something around 67. All the codes and constraints.
And that's if the gene is labeled. If you go online and you just are looking at a genome sequence, you know, it starts with the beginning of the DNA molecule, and that's one. And then you just have, you know, random numbers from there. Or it's-
Where it's positioned is
So promoter will always be upstream. And that helps you to determine whether the gene is on the top strand or the bottom strand. So if, kind of like some of the problems that we saw previously, but if we just saw a promoter and we knew that RNA polymerase is moving in this direction, then we know which strand to use, right? Because it has to read from 3' to 5', so it has to use the top strand. The gene is downstream of the promoter. Right, it's always after the promoter. Was there a question in there? All right, so why don't we work on these captains? So, correct. And think about how that makes sense because the RNA polymerase has to start at the promoter. So it has to be upstream of the gene.
We talked a lot about RNA polymerase too. Because you said it's in women, do we have to know like the differences?
No. So I'll give you guys probably 20 minutes to go through these, then we'll see where we're at, see what we need to discuss. And you can work with the person next to you, it's fine. You refer to your notes.